things. If not, it's no big deal. We can just do a massive one tomorrow. Because tomorrow all we'll have to talk about is vision and hearing and stimulants, which definitely we'll finish that easily before the break. So we'll have a huge chunk of time for all of our lab stuff. Let's talk about taste first. Let's see. Review of the tongue and different types of papillae, right? And we had some papillae that are just bumps, really, to give us the texture. And then we had our other types of papillae that actually are going to have receptors for taste. So they just happen to pick. Oh yeah, it's faded out on that. But they happen to pick one of the circumvallate ones and then make it bigger. Doesn't matter. Either one is fine. And now we look at this and then kind of look into this pit right over here that's kind of around that papilla, and we get our extreme close-up of these items, which are the actual taste buds, right? So let's do the big close-up here, and we've got our epithelium. Woo! Epithelium. We've got connective tissue supporting that epithelium. Woo! Anatomy review. And embedded in the epithelium is where we have our actual receptors, right? Those sensory receptor cells here for taste. They're going to be the taste buds. Taste buds. So, like each one of, see this bracket here? So each one of these, right, is the taste bud. It's kind of like a flower bud, right? Like a little rose bud or something. So maybe that's a way to remember that each one of these is a taste bud. And if we kind of really look up close, each taste bud seems to have multiple cells, right? And at the ends of the cells that are the actual, receptor cells, we have, I don't know if you can see this, I hope you can see this, these little tiny black lines are taste hairs. Taste hairs. So we've got taste buds made up of clusters of sensory cells, receptor cells, and at the ends of the receptor cells, we'll have taste hairs. Taste hairs. Okay, so far so good. Oh, go ahead.
I'm just curious, so when you have that painful bump on your tongue, what is that from?
A lot of times it's like maybe when you bite your tongue or something like that, and then it like swells up. Is that what you mean?
Somebody told me it was a dying taste bud before, but I don't. You know, when you have a painful bump, it happens to everyone, I think. Hopefully.
Morgan? Continue. I'm not sure. I haven't heard that one. Like automatically I went to like, oh, like when you bite your tongue. I just haven't heard that. Maybe we could do a search on it and see like what's out there. And maybe what, narrow down a little bit. I don't know. I think it's worth it.
Okay.
Trying to narrow it down. We'll find it. We'll see. We'll see what it is. All right. But anyway, for this, let's see. We left off with the hairs, right, at the ends of these receptor cells. All right. Because we're trying to sense stuff that we're eating, right? The stuff that we're eating is going to be dissolved in the saliva, and those are all chemical things, right? So we're dealing with chemoreceptors embedded in these taste hairs, in the cell membranes of these taste hairs. So, first, I guess, before we look at that next picture I was thinking of, here are our categories for different types of things that we would taste. So we'll start off with maybe the easiest one for us to understand based on what we know, sweet stuff. Sweet stuff is all sugars. And we know all about sugars, right? Carbs and especially glucose. We're all over that. Wow, awesome. So glucose, obviously ligand-gated ion channels, right? It's going to bind to ligand-gated ion channels in the cell membranes of those taste hairs, open up, right? And in this case, like so far, I'm going to come back to that, but we know all about like, oh, that would create a graded potential, right? That kind of stuff. So let's narrow it down, or maybe add, I should say add to it. Remember we've mentioned, I think maybe twice so far, something about second messenger pathways, and we saw one of our examples was like GMP. Remember we had like, no, I think we haven't got to GMP yet. We've had GTP and GDP, I think is what we've had. We'll see that Thursday. But remember we said, oh yeah, we could use that, and maybe that was this extra step, right? And then we also said that we could trigger like an enzyme cascade inside the cell, and I think the example that we saw last week was to trigger the cell to make more receptors, right? And then we were going to embed all those additional receptors into that membrane so that the next time that stimulus comes in, we've got so many more receptors that was going to help us to process that information faster. That was like learning, right? And remembering information. So that was an example of a second messenger pathway, right? Some signal coming in, opening up a channel, and then by that having that channel open, okay, we not only create our graded potential, but we also trigger a cascade, right? That's going to do this whole other thing that will help us. All right, so yeah, we've got our sugar. Yeah, we've got our ligand-gated ion channel. Yeah, we're going to create our graded potential, right? But we're also going to trigger this second messenger pathway, right? That is going to block potassium channels. I mean, everything's all, right, it's all in the same spot. Potassium channels. And we end up with our depolarization by blocking hyperpolarization. Does that make sense? So it's a little bit of a roundabout way to create our graded potential. Good? By blocking IPSP, we create EPSP. Right? Kind of coming in through the back door. All right. Sweet stuff. Let's see. Sour stuff. Sour stuff tends to be acidic, like lemons, right? So if something is acidic, we've got a lot of hydrogen ions. We know that. And it turns out hydrogen ions can block potassium channels. So kind of the same thing, right? By blocking hyperpolarization, you end up with depolarization, right? All right, but the hydrogen ions are going to just sort of directly block the channels, potassium channels. All right, I think that's rather straightforward. Let's look at salt. Salt, of course, is NaCl, right? Sodium chloride. So when that dissolves in saliva, it's going to dissociate. You've got your sodium and you've got your chloride ions. So sodium ions, I mean, might as well, right? Just like enter into a sodium ion channel. Just be really straightforward about that one, right? All right, so that's pretty good. We know that, right? We know that stuff, so that's pretty straightforward. Bitter things. I mean, it depends on the person. Some people like bitter things, or a lot of people like some bitter things, but not all bitter things. But a lot of bitter things happen to be poisonous things. So we definitely include that on the list as something good for us as humans to perhaps dislike, right? Especially those poisonous ones, because then obviously you would not eat it anymore, and maybe you'd actually spit it out. Maybe it's like totally gross. All right, so bitter things are going to work using second messenger pathways. We're not going to go into the details, but we'll know, okay, second messenger pathways, maybe some enzyme cascades leading to depolarization of those particular hair cells, right, or taste hairs, and then those cells, right? And then we've got our umami or savory. That has been relatively a recent addition, but that's been here for a while anyway. But also second messenger pathways. We're not going to go into all the details of that. Good, and depolarization. So this is as much detail as I want you guys to know for our level of understanding for taste. I think this is great. Reminder, but this is a rather strong reminder. I know this is anatomy, right? So we had innervation of the tongue. Woo! Cranial nerves, right? Oh man, be sure that you remember not only the proper name of each cranial nerve, but the Roman numeral way of naming, right? The cranial nerves that carry those various graded potentials that we generated at the taste buds, right? Carry that information to brainstem, and then up to thalamus, and then gustatory sensory cortex. Good. So review, right? Review. Be sure that you've got this one, right? Because that's kind of our big overlap between anatomy and physiology. Putting it together, right? All right. So that's it for taste. Taste. We're going to go kind of light as far as information and depth goes. Smell also. We're going to stay on the light side. We're going to go nuts with vision. And a little bit nuts with hearing and vestibular. We're going to go crazy with vision on Thursday. So, but let's be grateful for this part. All right. So let's see. Here we've got smell or olfactory stuff. We remember the anatomy. Be sure you remember that. And let's focus in on that olfactory bulb sitting on top of what bone? What part of what bone? Holes in it. Let's see. Let me see if I can get this. Isn't it ethmoid? Ethmoid bone. Okay, good. Did you say that one in here? Oh man. Cribriform plate. Ooh. Okay, so cribriform plate on the ethmoid bone, right? That top right? Darn. Oh, so sad. Okay, sorry. Okay, so ethmoid bone. But so you have cribriform plate on the ethmoid bone, right? And holes in it. All right. So we see little nerves, right, from our olfactory nerve poking through. Or actually, I guess it's this way, poking through those holes. And let's see. Here's our close-up of that. Here's our receptor cell. Good. Sort of as a part of this epithelium, right? That's covering that cribriform plate on the nasal side, right, inside the nose. And we'll have a layer of mucus here that was produced by sort of the rest of the epithelium, the mucus part, right? Mucosa there. And we have some cilia. These particular cilia are not the ones that are responsible for pushing, right, moving the mucus. These particular ones are just there to grab, right, molecules that are dissolved in the mucus, the odor molecules dissolved in the mucus. All right, very good. So here's just a simple explanation. Molecules, whatever they are, get dissolved in the mucus. They're going to bind to receptors, right, embedded in the membrane of these cilia, right? We'll use some second messenger pathways. I'm not going to go into the details, but as always, open up ion channels, depolarization, right, and there's your graded potential. Good? Or receptor potential. And we send that, here's olfactory nerve. Ooh, awesome. What cranial nerve is that? Because it's not up here. Good review. Cranial nerve number? Let's see. Oh, wait, I didn't have that review. Why not? Well, I'm sure it's got to be here. One of these review slides. Ah. Okay. Just remember, olfactory bulb, right? Olfactory nerve, or you say tract. That's fine. This is the one exception to our rule of having two synapses in the thalamus, right? We don't have to go there. We can go straight to primary olfactory cortex, and we know from anatomy that that is incredibly close to limbic system structures, right? So smell is very, very, very intimately tied to our memories, right? We have hippocampus, right, and amygdala and all of that in the limbic system. So very, very closely tied to memories and the emotional component of those memories. Good. All right. So, all right. I think that finishes out the smell stuff. How are we doing on time? So, this does bring us to the light section. Let me do this little intro, then we'll be done with lecture, and let me show you what we're going to do in lab, like as far as like the pages in the book and stuff like that, so you can look at it ahead of time, because it's like instructions, like how to do this and how to do that, right? So you can a little bit prepare, and then we'll be done. I'm sure we'll finish early. So let's see, just a super quick intro into vision stuff. Got cut off, but these are all waves, right? Waves. And our eyes are geared to be sensitive to this tiny little bit, right, of wavelength in here. And this would be known as visible light, at least visible to us. I mean, other animals have different, right, sort of amounts of visible light that they are sensitive to. So this is just focusing on humans. Don't worry about the individual things like, oh, this is yellow and this is blue and, right, all of that. Let's just know in general, humans, it's pretty narrow, right? And rainbow, very good. All right, so it's good enough for us. Let's see. And be sure that you review your anatomy of the eyeball, right? So it's pretty much all here, right? What you need to review for anatomy. Definitely, when we do the lab on Thursday, there are a whole bunch of things to do for vision. So it will greatly behoove you to have the anatomy fresh in your head for the eye. Because I looked, we do have pretty cool things so that we will be able to look. Hopefully, I don't know, I mean, I haven't looked myself, so I don't know exactly how good they are. But in theory, we should be able to see the retina. We'll see. We'll see what happens in real life when we actually use those, because I haven't used them yet. But anyway, review this anatomy, and review this, right? This is the surface of the retina. Good. Keep in mind, I wish they would have flip-flopped it. Let's see, because this, I added this in the other day because I noticed it and I was like, oh, this is a left eye. So I added in, right, this is a left eye. And this is a right eye. So, I don't know what to do that. But anyway, and here, let's see, this should all be review. Maybe I'll talk about this. But this is, some of it's review of anatomy. I would like to focus in on, so be sure you understand this that we'll talk about now so that Thursday is going to be good. Let's talk about this. Ciliary muscle. Ciliary muscle. So we see in our anatomy picture, remember that? Ciliary muscle was going to control the shape of the lens. Good. And the way it works is counterintuitive. So when the ciliary muscle is relaxed, because it's a circular muscle, in this picture, I think it's very deceptive when they give you this slice of the eyeball and then you see, oh, ciliary muscle. And it appears to be a muscle that would approach the lens like this, like kind of coming at the lens. But it's not that way. It's really a circular, more of a sphincter-shaped muscle that will tug on these fibers, right? You see these white zonular fibers? So that circular muscle is coming around this way, and those zonular fibers are the ones that are, right, going to kind of come at the lens. But because the actual ciliary muscle is arranged like a circle, when it's relaxed, you see how, okay, oh my gosh, I don't know if I can do this. You see how this is like a circular thing, and there would be the fibers coming off of it, right? And it could tug on the fibers, right? So if the muscle is relaxed, that's when it's actually tugging on those fibers, like this. So a relaxed ciliary muscle is like this. Let's say a contracted ciliary muscle, we know about sphincters, right? So contracted would be smaller, and relaxed would be bigger, right? That's in general for a sphincter. So think of ciliary muscle as a sphincter. Good, so relaxed, contracted first. That's our first thing. So if we've got these fibers that can pull on the lens, right, that go from this muscle to the lens, right? If those fibers are taut, right, they're being tugged on, can you imagine that you would flatten the lens, right? And if those muscles are loose, right, the fibers themselves, not muscles, those fibers are loose, the lens would plump up, sort of swell up, you could say swell up, I guess, right, and plump, right? So, when the ciliary muscle is more like this, you see how that would pull on the fibers, right? So the ciliary muscle is relaxed, but the fibers are taut, right? So what is the lens going to be? If the fibers, it's going to be more flat, right? Because the fibers are kind of tugging on it. But see how it's counterintuitive because the muscle is actually relaxed as a sphincter. It's bigger, right? Okay, so now when we contract down on that muscle, right, see how now it's got like a smaller circumference, but what happened to the fibers that are going from this muscle to the lens in the center? The fibers are loose, right? We've released the tension, right? We've sort of slackened the fibers, make sense? So what can happen to the lens when you release the tension? Rounder, exactly. So contracted ciliary muscle, we release the tension, right, on the fibers, the zonular fibers, and that allows the lens to, right, to become rounded. Good? So it's counterintuitive. Be sure that you really understand that because that's going to factor into our lab on Thursday. All right. Let's see. Also, let's remember parasympathetic stuff from anatomy, right? We must begin to know, starting now, we must start to drill, right? Drill into our heads all of the sympathetic and parasympathetic. We're going to focus on parasympathetic and sympathetic. We're going to focus on parasympathetic now. But we've got to start like getting those things like seared into our heads, right? As we go through each of the organs that have parasympathetic and or sympathetic innervation, oh man, we're going to have to really super focus on that because that, remember the scary chart? That scary table? That was all like alpha and beta. Oh man. That was that, right? So we have to start now. We're going to go back to that table. And we're going to see. So, ciliary muscle was under the influence of parasympathetic. Oh my gosh. Parasympathetic stimulation. So, if we just went through this whole thing, right, of relaxation and contraction, right, relaxation and contraction, and having a flatter lens or a rounder lens, right, that, right, shape of the lens is under the control of, well, parasympathetic system. Good? So far, so good? All right, we got to start putting that together. Wait. Hang on. I'm going to call it here. Let's see. Yeah. I think we'll save scary stuff for tomorrow. But feel free. Oh my gosh. Yeah, feel free to look through that. It's a really big topic, so I want to give us like a good chunk of time. But I know I can ask you this, and then I want to look at the lab stuff. What cranial nerve? What cranial nerve is going to carry this information oculomotor, which is cranial nerve number? 3, almost. 3, right? Good. That was good. What information is carried through cranial nerve number 2? And also, what's the name of cranial nerve number 2? Optic. Optic, and that's going to be what information? I mean, that's tomorrow's topic, really. But maybe remembering from anatomy. Sensory, yeah, like just regular vision, right? Just vision, right? Just light. Plain old vision. So, so let's start to get that anatomy back, right? Because tomorrow we're going to be all over this. It's a huge topic. So, cranial nerve number 2, right? Optic nerve, vision. Wow. Cranial nerve number 3, oculomotor. Actually, in tomorrow's lecture, we're going to talk a little bit about moving the eyes, so we'll talk about that. But also, we're going to talk about this parasympathetic component, right, of cranial nerve number 3. So take a peek back at that stuff from anatomy, good? And this eye anatomy, and then you'll be in a good place for tomorrow so that we can really have a big discussion, good? And all this stuff. So that'll be enough for a lecture for today. Just so you know what to be prepared for lab. The edition that I happen to have here today with me is the 13th. I know there's a 14th edition out there. It's okay, either one is perfectly fine. Like I said, it's also fine if you have a used one, and maybe if the little work pages are ripped out and already turned in, it doesn't matter. I'm not going to have you guys have fresh pieces of paper. So bring in several slices of fresh paper. Doesn't have to be graph paper because we're not going to do any. We're not going to do any graphing. I don't think we're going to do any graphing. We'll be making charts, right?